Hello, my name is Michael Johnson. I'm the CEO and one of the co-founders of Visical. Today I'll be talking about identifying the ideal in vitro liver model for various research applications and how we can apply different liver models to answer different types of research questions. Now, the overview of this presentation starts off with some background on Visical. Who is the company? Where do we come from? What do we do? I'm going to dive into an overview of various liver in vitro models, what they're used for, why they're important, where the field is currently. And then I'm going to dive into three different examples: 2D liver cell culture models, focusing on the Hirel microliver platform. I'll discuss 3D liver cell culture models with a strong focus on NASH and drug-induced liver injury. And then lastly, I'll touch upon tissue slice ex vivo models, so actually using slices from real in vivo tissues as an in vitro model. Now, to kick it off with some background on Visical. Visical is a company that I founded as a spin-out from Rutgers University about 6 years ago. And we are an industry leader in advanced models and tissue imaging focused on improving and accelerating drug discovery and development through contract research services. So we operate primarily as a contract research services provider, but also sell a few reagents, kits, and also liver cell culture models through the Hirel platform. We have a worldwide customer base. All 20 of the top 20 pharmaceutical companies in the world are clients. And we also, of course, work with medium and small-sized pharma companies as well as other prestigious research institutions and non-governmental organizations. We have an expertise in drug discovery, assay design, oncology, immunology, inflammation, and of course, liver disease and liver disease modeling. The two focus areas for us where we play as far as services are advanced imaging and advanced cell culture. When we started our company originally about 6 years ago, the focus was squarely on advanced tissue imaging. But after a short while, we had a lot of researchers coming to us to image 3D cell culture models, organoids and spheroids in 3D using unique confocal and light sheet technologies that we had developed. And from there, we decided to build out our own advanced cell culture capabilities and end-to-end assays involving advanced cell culture models. And of course, in that space, the high focus area initially was liver, and now we've expanded as an industry to lots of other models, which I'll not touch upon today, but of course, there's lots of other 3D cell culture models that are out there that are now widely used in academic as well as industrial settings. But for us, we do 2D and 3D cell culture models and assays, as well as ex vivo tissue slice assays with a strong focus on high content imaging endpoints. And we do a lot of work on custom model development if we don't have a model or we don't have a model from a third party that meets the needs of our researchers. We were acquired back in May of 2021. So after being a standalone company for a little over 5 years, we sold Visikol to Bico. Bico is a Swedish-based life sciences company that is a family of 15 different companies that are broadly focused on this concept of bioconvergence. Essentially, we operate as a tool, reagent, consumable, equipment company that's trying to facilitate life sciences research with a strong focus on cell culture, robotics, automation, dispensing, and other technologies which help researchers accelerate their drug discovery and development processes, as well as basic and translational research. And for us as a company, what we're doing around this bioconvergence concept is we're taking a bunch of different technologies and combining them together to accelerate the time it takes to get a drug from the lab all the way to a patient. So for us, we're very focused on advanced cell culture assays and models, which I'll talk about today, advanced imaging tools, and then also AI and image analysis. So we generate huge amounts of data as a company through our high content imaging and through our 3D cell culture assays and 2D cell culture screening. And we use advanced tools for data processing to turn that into something useful that allows us to make more informed decisions as part of the drug discovery and development process. Now, for liver cell culture, when we think about liver cell culture models and assays, there's a lot of unmet needs that we're working towards addressing. Liver toxicity is one of the most common sources of late-stage clinical failure for therapeutics and necessitates more clinically relevant in vitro models. We need to bridge the gap. If we're able to better predict toxicity in inexpensive and high-throughput in vitro assays, we're better able to avoid late-stage clinical failures that result in longer time to get therapeutics to market, and of course, also higher prices for therapeutics for patients, which we want to avoid. So we're trying to shorten that time by better predicting what's going to fail. Liver disease, so NASH, NAFLD, and HBV are of the most common diseases out there in the developed world and affect tens of millions of people in the US alone. And trying to develop in vitro models that mimic these help us better understand these diseases and also develop therapeutics to these unmet needs. Where we operate as a company is we have several different liver cell culture models, which I'll dive into today, that have different advantages and disadvantages depending upon the research context you're working in. Now, as I touched upon in the last slide, when we look at drug-induced liver injury and late-stage clinical failures, a lot of this is caused by toxicity, and hepatotoxicity is the #1 toxicity that we're seeing in the late stage that's causing these failures. And of course, by reducing that in late stage, we're able to improve this funnel of developing new drugs and reduce the overall time and the cost to do it. There's several other toxicities which are leading causes as well, but liver toxicity definitely takes the cake, and also disease modeling for NASH and other disease etiologies that I mentioned. The focus of this presentation, though, is on which model is right for your research question. There's a lot of researchers that are looking at organs on a chip and bodies on a chip and these very complicated models, and we see these papers come out about the end of animal studies. And of course, we're trying to reduce animal studies, we're trying to reduce human studies, and get drugs to market more quickly. But when we look at in vitro systems, there's never going to be a model that answers all of our research questions. Or earlier in drug discovery, we want higher throughput and lower cost. When we're later in the process, we want higher relevancy. The price is going to be higher, the throughput's going to be lower. So there's always a balance between throughput, price, and relevancy, depending upon where you are in part of the drug discovery process. And I'll touch upon several models today, but when we look at 2D and 3D and ex vivo, they have different characteristics. Some are really relevant as far as mimicking the microenvironment within the body, while others are much less relevant but much higher throughput. So a lot of where you're picking a model and what model you pick is based upon which of these characteristics you need, but no model is going to meet all three of these parameters at the same time. We as a company at Visical, we have a broad spectrum of liver models because, again, we recognize that no model meets every need. So at our company, we have a broad range of hepatotox, DMPK models, as well as looking at mitochondrial, metabolic, immune cell infiltration, and efficacy within liver models, and also modeling liver diseases like NAFLD and NASH. And we have this spectrum, which I'm showing from less complicated to more complicated on the far right, with our 2D cell culture model system being the Hirel microliver model system. This system is primary hepatocytes. It's 10 different species. And we actually recently acquired this platform back in September of 2021 and integrated it into Visical. And this has allowed us to develop the most broad liver portfolio of in vitro models in the marketplace, stretching from 2D all the way to ex vivo models, which I'll touch upon in a moment. We, as a company, also have simple spheroids. So spheroids allow us to have the complex three-dimensional structure that we see within the body in a format with a little more complexity than 2D. It allows us to answer different questions than we could in 2D. We also have complex spheroids as a company, so multiple cell types combined into a single model, as well as iPSC-derived models. And then lastly, ex vivo tissue slice models are when we take a tissue, an actual liver in this case, we slice it into about 200 micron thick sections. We put them on transwells and we keep them in culture for a period of time. We can do that with healthy tissues. We're inducing NASH. We can do it with healthy tissues. We can also do it with NASH tissues, for example, and actually look at the amelioration of the disease in actual human tissue, but in an in vitro format. So we're taking one tissue and turning it into lots of different experiments through the ex vivo platform, which I'll talk more on later. But again, these models are going from less complex to more complex on the far right. The trade-offs are throughput and cost, which I'll touch more upon later. So not every model is great for every research question. I'm going to dive into a few of these examples in more detail and also touch upon which one might be right for your specific research question. The first one I'll dive into is 2D liver cell culture. This is the one that we're most familiar with. As of 5, 6, 7 years ago, most folks were doing 2D cell culture work or just suspension work. And as we've shown that models become able to answer questions better than 2D and suspension work, we've started to shift over to 3D and other more complicated models. But for a long, long time, those were our primary models that we used, 2D and suspension, especially in the hepatocyte space. And the model that we've integrated into our services and we've acquired recently is the Hirel microliver model. So unlike a lot of other 2D cell culture platforms that are out there, this unique system allows us to have long-term metabolic stability compared to just plating hepatocytes on a simple plate like most researchers have done. This platform has 10 different species, so we're able to look at not just primary human hepatocytes, but also dog and cat and mouse and rabbit and a few others as well. It's been published in over 100 publications to date. It's compatible with standard automation and analytical equipment, and it's used by most major pharmaceutical companies for DMPK work as of today. So it's a very unique platform, really unique technology. And the rationale behind it is that we were, as a company, as Visical, looking for a really strong metabolically and phenotypically stable model. And Hirel was able to have metabolic and phenotypic stability for over 4 weeks in culture and sometimes much longer than that, and also able to sustain infectious diseases like HPV, which makes it a really unique 2D cell culture platform. The format for Hirel microlivers is just standard 24, 48, 96 well plates, and we're able to put several different species on a singular plate and sell it as a product if researchers actually want that. But again, the strong thing here is for any of these species that we're working with the unique Hirel cell culture approach, we're able to get over 4 weeks stability and metabolic activity, which again, we just don't see in traditional suspension or traditional 2D cell culture, which allows us to look at low clearance compounds in a really substantial way compared to other models that are out there. I mentioned the phenotypic stability. We have published several papers on this, but as far as comparing to other models, it's just not even close. Phenotypic stability of the Hirel system is the gold standard in the space as far as long-term, long-lasting stability, especially compared to 3D cell culture models that are out there. And for metabolic competency, it has the highest sensitivity and predictive capability in the space compared to other 2D, compared to 3D. And a lot of that is due to just a large number of hepatocytes in the system. That also is why it's so helpful with looking at low clearance compounds. But this Hirel microliver model platform is really ideal for metabolism studies, PK, PD studies, and really is just a tried and true approach for looking at those. We as a company didn't have a model that filled the space, which is why the acquisition of Hirel fit in so nicely to what we're doing. It's a model that meets this use case incredibly well, and also provides us a lot of flexibility with species. So if we have a result or we have a unique finding with rabbit, for example, or with monkey, for example, we can go into the Hirel system and on a single plate compare multiple different hepatocytes, species hepatocytes with each other and see if that was species specific or if we see that in multiple species. And we can look at the underlying metabolism and metabolites as well and see exactly what's going on. So really a great model for metabolism. The next one I'll dive into is 3D cell culture. Now, 3D cell culture has evolved dramatically. 5-6 years ago, when we were at conferences, Society of Toxicology and SLAS, this was primarily a lot of academic researchers. And then really rapidly, every pharma company, every biotech company started to rapidly adopt 3D cell culture, initially as spheroids, and then as organoids, and now looking at much more complicated systems. But the rationale is that for a lot of research questions, 3D better mimics what's happening with the body. It better answers our research questions and allows us to answer unique questions that we couldn't otherwise address. For example, if you're trying to understand how a large molecule drug penetrates into a tumor, for example, of course, out of this context, the 3D cell culture environment allows us to look at spatial context, allows us to look at infiltration, penetration, and also cell-specific things like if we have a T-cell assay, where are those T-cells going? Are we getting T-cell killing? Where is it? Is it on the outside, the inside? So we're able to answer more complex questions and add a spatial context to the biology and to the research. Our body is, of course, in 3D. It's not in 2D. It's not growing on a plastic plate. And therefore, having a 3D cell culture model starts to better mimic the body and allow us to answer more complex questions. One of the models that we use regularly at Visical is a primary human hepatocyte spheroid model. So very simple, just primary human hepatocytes in a model. It's affordable for dye screening. It's in a Corning ULA plate, so we're able to have it compatible with high throughput screening if we want to have imaging as our readout. And typically these are about 200 to 300 microns in diameter. Going much beyond that, the problem you start to have is, of course, the inside starts to become necrotic. Imaging becomes a little more challenging as light penetration, refractive index mismatches prevents you from imaging the entire structure. So we stick to about 200 to 300 microns in diameter for these models, but typically do it in 96 or a 3D4 well plate format. And when we look at large screens of compounds, what we're able to show is compared to 2D models, we do have a higher sensitivity. Of course, this is a dye assay, so looking at drug-induced liver injury. But compared to 2D monolayer, we get a better ability to look at false negatives, false positives, and overall just a much more sensitive assay in this circumstance compared to traditional 2D. So again, not great for all research questions. When we look at something like metabolism, we're not able to look at low clearance compounds. We're not able to have the same type of sensitivity in that respect that we do see here for DILI. So this is a really unique model for DILI, and it's also in a high throughput format, which makes it really ideal for screening large numbers of compounds. And when I say large, I mean several hundred compounds at a single time. And the next model I'll talk about is just a simple model, so HepG2 spheroids. So cancer cell line here, we're looking at a hepatocarcinoma, and we're able to look at something very simple, so looking at steatosis. So we can use the same simple 3D cell culture model. Instead of looking at primary hepatocytes, this time we're looking at HepG2, and it allows us to look at something as simple as lipid accumulation over time. The reason I show this is because what we're showing here is lots of different models for lots of different research questions. HepG2 is different from primary hepatocytes in a bunch of respects. And one of the main respects is that it's HepG2 is a cell line. So we can use continually the same sort of model, the same kind of thing we see with HepRG, whereas we work with primary hepatocytes, they vary batch to batch, they vary donor to donor. If we pool donors, we don't know exactly what we're getting unless we do really thorough characterization. So if we need a high level of reproducibility, we don't need that much complexity, we don't need primary hepatocytes, perhaps something like HepG2 or HepRG becomes a little bit more appropriate. So again, there's other factors involved, but not every model is perfect for every application. Then diving into the next one, we call this model open liver HepRG-NP. And what this is it's HepRGs with non-parenchymal cells. So we have several different cell types in a spheroid model. And in this model, we're able to look at, because we have those NPCs, indirect immune-mediated DILI. So again, if this is a question we're going after, we need a slightly more complicated model than just HepG2 cells or just HepRG cells in a spheroid. So we're basing the model on the research question instead of the other way around. We're not trying to force a model into a research question. And then a model like that can be used for other research questions as well, like looking at NASH. NASH and non-alcoholic fatty liver disease are things that we don't hear a lot about, but these affect a large proportion of the population worldwide. We look at non-alcoholic fatty liver disease. This is essentially due to poor diet. Poor diet is, of course, ubiquitous in the developed world, and the prevalence of this is incredibly high. So when we're talking about non-alcoholic fatty liver disease, we're talking about the accumulation of lipids in the liver, inflammation, fibrosis, and eventually in advanced stages of the disease, the liver starts to shut down and not perform how it's supposed to. So a very serious disease. The disease process takes an incredibly long period of time. This is a decadal long disease process. So of course, modeling it in an vitro setting in a few weeks is challenging. But as a company, we're trying to develop models that help researchers better understand this long-term process. Can we slow this disease progression down? Can we reverse it? What can we do to actually improve this process here? So a model that we've developed for this, which I alluded to before, is this HepRG and NPC model. It contains hepatocytes in the form of HepRG cells. We have our liver sinusoidal endothelial cells. We have Kupffer cells, stellate cells, cholangiocytes. So most of the major players that you see in the liver are represented in this model, and that allows us to mimic the disease state of NASH in this spheroid context in the same size, about 200, 300 micron diameter spheroid. And with this model, we're able to show glycogen storage, transporter protein expression, P450 enzyme expression. And with NASH, we have several assays that we've developed, but just looking at high content imaging data here, we're able to look at things like collagen deposition, alpha SMA, of course, live dead, PDGFR beta, and understand how different compounds might ameliorate this cascade, how they might slow down collagen deposition. And it's a really strong assay. We've run a lot for researchers in the space. We also, of course, do qPCR and ELISAs and other readouts as well, but the images tell a great story here when we look at them. So this is an assay that's amenable to a lot of different endpoints, but as a company, we do a lot of imaging, so you tend to see a lot of imaging data coming out of our lab as a big focus of a lot of our assays. So again, we're tailor-fitting the model based upon the research question. For this question, we need our NPCs. We can't just do it with HepRGs. So we're using a slightly more complicated, slightly more expensive model to answer this research question than we would for some other research questions. And then, yeah, looking at the next one, again, so same HepRG NPC model, and we're able to show here with just two compounds that folks are looking at today, elafibranor and pioglitazone, what happens with different concentrations over time as far as looking at collagen deposition and alpha SMA. And what we're able to show is, again, this model is very reproducible, works very well. We have a pretty big assay window with our controls, and are able to evaluate compounds in a very meaningful and accurate way for our clients. And then next one here is pancollagen and alpha smooth muscle actin analysis. Again, looking at these same compounds for elafibranor and pioglitazone. Fibrosis was induced in these, and we're able to look at that. We're able to quantify it, and the assay window again is pretty large for this assay, which makes it quite powerful for the researchers that are looking at different compounds and their effect on, of course, collagen deposition and just generally the NASH cascade. The next one assay we'll look into, and next model type, are precision cut tissue slices. In this case, PCLS, precision cut liver slices. Precision cut liver slices are an alternative type of in vitro model for evaluating antifibrotic agents as well as other research questions. We, as a company, have three different approaches. We have normal human tissue, we have diseased human tissue, and we also have mouse diseased tissue. In our normal human tissue, we can induce NASH, or we can just use them as normal human tissue. We've also done some work with HPV human tissue as well. The idea behind this model is that we take just tissue shortly after a patient is deceased or shortly after an animal has been sacked, and we take the tissue, in this case liver, we section it using a compressed tome from a company called Precisionary into about 200 micron thick hockey pucks. We put those on transwells, and we're able to keep those in culture for anywhere between 2 and 5 days, depending upon the type of tissue that we're getting. Some tissue is, of course, healthier than others, and rodents are different than humans, of course. But for these types of assays, usually we're looking at about 400 slices. We're looking at multiple time points. We're doing ELISAs, we're doing qPCR. We can also do high content imaging and histology as well as readouts. We tend to do a lot of readouts from these assays just because getting a human liver is quite expensive. We want to make sure we get out as much as possible. But this is a really unique assay because we're taking the native tissue architecture, we're taking the native tissue itself and using it as an in vitro model. So you're working with real human tissue and keeping it in a culture. The problem, of course, the downside of this model, especially in regard to NASH, is that we're trying to recapitulate a disease process which takes 20, 30, 40 years in a model in a few days. So of course, the findings that we have are not going to be one-to-one. The concentrations are not going to be similar, but what we're seeing is that it gives us a really good model of what's happening with the real disease, the real pathways that we're looking at within the body. So again, it's a model, so not perfect, but we've seen great success with this so far with our clients, and I've run dozens of studies so far with this PCLS format. To look at the mouse study, we usually use mouse CCL4 models. So we're acquiring the tissue, we're generating the slices, we're keeping it in culture. And the CCL4 model is from a partner company of ours called Melior Discovery in Pennsylvania. We don't have pre-culture. We treat for 72 hours with an alk5 inhibitor. And then what we're looking at here is on the top right, PDGFR beta and alpha SMA with DAPI in blue here. And we're able to look at the untreated versus treated and see how alpha SMA area increases. We can quantify it by a bunch of different ways, but with high content data, we're looking at, in this case, area. We can look at volume for collagen deposition or total collagen deposition. And also in parallel, we're looking at qPCR and other readouts as well. We're able to show that it's, again, a very powerful assay with a really strong assay window for looking at compounds that hopefully are going to ameliorate NASH or reverse the disease process. For the normal human PCLS, we can stimulate the disease state. So we're looking at F0 and F1 human livers, pre-culturing for 5 hours and then stimulated for 48 hours. And then typically we're doing free fatty acid stimulation for inducing collagen deposition. And then we're quantifying again with the same histology, high content, or qPCR/ELISA readouts from this assay. The benefit of this, of course, is it's human, so it's more relevant than looking at a rodent model. But the disadvantage is, of course, we're beholden to when we can actually get tissues, and the tissues don't always meet our requirements. We have to have a very specific set of requirements for our assays, but of course, we don't always know all the information about our patients. So it's a little bit challenging to actually identify that the model meets our specific needs. but we've demonstrated a strong ability to get livers, at least several per month, of human healthy livers for these studies. But in this case, we're inducing disease states. So that's another disadvantage in the context of NASH compared to actually getting a NASH tissue itself. Now, we can get NASH livers, which is what I'm showing here. And of course, visibly, a NASH liver looks very different than a human healthy liver. The problem with NASH livers is they're harder to get. And one of the reasons for that is we don't always know the true medical history of a person. It's hard to get a person who has NASH but has never had an alcoholic drink in their life. So trying to balance the two is always an issue. And these come less frequently, of course, than healthy human livers. So it's a powerful assay in that we're not only getting the human tissue, we're getting the human tissue with the disease state that we're trying to treat, and when we're trying to look at ameliorating the disease state in the tissue itself. So a couple of advantages. Disadvantage, of course, is the tissue availability and meeting the specific criteria that we have. There's also disease state variability. So some NASH livers are very different from other NASH livers. We can have patient data that tells us that a patient's really unhealthy, and we get the liver out, and it doesn't look that bad. In other cases, it's a complete surprise. So we don't always know exactly what we're going to get. There's a lot of variability from donor to donor for these studies. But the question remains, what model is right for you? When we're developing a research project, we work really closely with our clients. We usually have several consultative calls at the beginning of the process to discuss the project. And what we're trying to find out with all of our clients is what is truly required. We see it as our duty to balance cost, throughput, and relevancy. We could give a client a really expensive model that answers the research question, but if it's not needed, we want to avoid it. So instead of using a HepRG NPC model, perhaps we're using a HepG2 model, or perhaps we're using just primary hepatocytes. So we're always trying to balance what's actually required to answer a research question, because we can always make something more complicated. We can put an organ on a chip, we can add microfluidics, we can add all these things, but if they don't actually help us better answer our research question and they're challenging to validate and they have other problems, why are we using that model? So for us, when we're looking at a model, and when any researcher is looking at a model, first question we always ask is, where are you at during the discovery process, and how many compounds do you have? If you have lots and lots and lots of compounds, you want high throughput, you probably want lower cost per N. And while relevancy is important and you always want to have as much relevancy as possible, perhaps you don't need an extremely relevant model and you can get by with a more simplistic system, perhaps a 2D cell culture model of just one singular cell type instead of a 3D model or a tissue slice model. The next question is, which model can mimic the disease state you're looking for? So there's lots of models, 2D and 3D, which will just not mimic your disease state. We can't get NASH if we don't have MPCs. So of course we have to have MPCs in a more complicated model. And there's several 3D cell culture models that will not become infected or not hold infection for a long period of time and stay viable. So the ability to actually infect a model, if you're looking at HPV, for example, is super important. So we want to see, what is the research question? Can the model actually address it? And then lastly, is there sufficient validation data to support your research question? Do we have an assay window in the model if we run it for your intended purpose? Do we have a lot of control data which compares the model in vitro to in vivo, where we can say that, yes, these results will be indicative of what happens in vivo? Because at the end of the day, the whole point of these models, these more complicated models, is that we want to better bridge the gap between in vitro and in vivo. And if we're not doing that, we shouldn't be using that model. We should be using something that's more traditional, more standard, and has a lot more data behind it. So these are kind of the key questions to ask. There's, of course, a lot of other questions as well. But what I really want to paint is that no singular in vitro model is going to answer all of your research questions. And in vitro models are not going away. They're not going to be replaced with some organ on a chip or body on a chip, at least for a really long period of time. And even then, they will always have a place. So in vitro models are great, as is any model, but at the end of the day, they are just a model. And you have to choose the one that meets the most, makes the most sense for your research question, cost, throughput, and relevancy. And with that, I thank you for your time. If you do want to discuss your in vitro program, if you want to dive into models in more detail, learn more about the assays we have here at Visical, talk about the Hirel models, please reach out to me directly. My email is michael.johnson@visicol.com, or reach out to us at info@visicol.com, and we can connect you with somebody from our in vitro team to discuss further. So I appreciate the time. And again, if you have any questions, please reach out.